So this is a lesson on different blood groups and what happens when you have a blood transfusion, how we know what blood to give you so that the body doesn't get attacked. So when blood is lost, your blood vessels constrict, and that's what we learned about last year with hemostasis. Bone marrow will then step up, blood cell formation, and attempt to keep circulation going. A whole blood transfusion, but that's not enough, right? A lot of times if you lose a lot of blood, you're going to need some sort of blood transfusion. In other words, you take blood from another human being and they give it to you. It is very routine when the blood loss is substantial. People have different blood types, and incompatible transfusion can be fatal. So we have to make sure that we know what each person's blood type is. So therefore, when you have a blood transfusion, or if you have to have a blood transfusion, I should say, then you don't get the wrong blood type. Therefore, it can lead to kidney failure. So blood cells. So all your red blood cells have either a glycoprotein on the outside or a lack of. So red blood cell plasma membranes contain highly specific glycoproteins. In this picture down here, these would be like your glycoproteins at their external surface, which identify each person as unique. These glycoproteins serve as antigens, which is a substance that the body recognizes as foreign if it is not its own. So if I'm type A, for instance, that means it has 8 antigens. I'll go more into this later, but then that means that my body recognizes these antigens, right? So it's like almost like a little alerting. If you're type O, you would have none whatsoever. So that's the only one that wouldn't have a glycoprotein is if you're blood type O. One person's red blood cell proteins may be recognized as foreign if transferred into another person with different red blood cell type. So if I'm blood type A and these antigens enter my body, the white blood cells are going to see it as foreign and attack it. You also have antibodies. So antigens are on the outside of your red blood cell, but you also have these antibodies as well. These recognizers are present in the plasma that attach the red blood cell surface that are different from those on the blood recipient. I have a really great table that it's on the back of your paper that tells you like what if you're A. So but really much if you whatever antigens you have, you carry the opposite antibodies. So if you're blood type A and you have 8 antigens here, you're going to carry B antibodies. So if the B blood comes in, it will attack it. Agglutination, this is actually what occurs. So if different blood type is transferred, the antibodies and cells will clump together. So this is the wrong red blood cell, right, with the wrong antigen. And the antibody will actually come in and it will attach. This will cause clotting, essentially, and that's what agglutination is. A few hours after foreign red blood cell is introduced, red blood cell is lysed. Most devastating consequences called kidney failure. This I added to the next slide, so don't worry if you see that's not there. But that's what agglutination is called, so we're going to get a little bit more specific in why this is bad for you. So transfusion so this is the reaction, right? So you've got the wrong blood type. This is back in the day, right? Maybe they didn't understand it. I don't know. Or you have a hospital that's just did like the wrong blood, gave you the wrong blood type. When the mismatched blood is infused, a transfusion reaction occurs in which the donor's red blood cells are attacked by the recipient's plasma agglutinins. So that is just those proteins on the outside of the red blood cell. First thing that's going to happen is a clog. So your red blood cells are going to clog small blood vessels throughout the body. So now and I'm just going to go back. So this is what we're looking at. So this is what all these little clots are going through your bloodstream now. So those clogs are going to start to clot your small blood vessels throughout your bodies because the antibodies attached. In the next hours, that clumped red blood cell is lysed or will rupture, and they're destroyed by your phagocytes. So those are your neutrophils and monocytes we talked about. So they're going to come in and see this as foreign, right? Because they're like, there's something wrong here. And they're going to destroy the red blood cell. It's basically, it destroys it from the inside out. The hemoglobin is then released into the bloodstream. Circulating hemoglobin passes freely in the kidney tubules. It's too big, and it will kill the cell, causing cell death and renal shutdown. If complete, the person will die. This is the most extreme. So these steps here are the most extreme situations. Like you got a lot of blood, they didn't know. It would cause kidney failure, acute renal failure. Other reactions, these things are not fatal, obviously, and it would be temporary until your body like got everything back to normal. But fever, chills, low blood pressure, rapid heartbeat, nausea, vomiting, and general toxicity. So this is just pretty much just reiterating why it's so important that we know what our blood cell types are, because this will occur. So if you get the wrong blood type, you can cause acute renal failure. So this is what it would look like in the serum. I'll talk more about this. You actually will do a lab that's like this, but so this is the serum. So this is your blood, right? So they take a serum and they put it in. So they'll take like anti-A serum, that would be like A antibodies, and they put it in, and if it agglutinates, then you know that it's that blood type. Okay, so because A would attack A. If it doesn't, so these are all agglutination. So this means that these are all clotting. This would be non-agglutination. So this means that it's not. This is important for your lab, what you're looking for, essentially. Like you're looking for that clotting look, right? That's what clotting looks like. This is what clotting does not look like. So blood groups. The groups are based upon antigens that are present. So type A, type B, or type O, or AB. If you're A, you just have the A antigen. B, you just have the B antigen on the outside of the red blood cell. O, you don't have any. And AB, you have both A and B. The antibodies formed during infancy against the ABO antigen is not present in your own red blood cell. So if you're type A, you would have B antibodies. If you're type B, you'd have A antibodies. So you have the opposite of what type you are. So if that blood type gets in it, then you're going to have that agglutination. Type O would have both A and B antibodies. And if you're type AB, you would have any antibodies because you could accept any essential blood. This chart is on the back. This is a very great chart. It will tell you what your red blood cell look like, right? So if you're type A, you have A antigens. What kind of antibody do you have in the plasma? Anti-B. The antigen is A, like I said earlier, and what are you compatible with? A and O. What would happen if you got B? The anti-B would come in and agglutination would occur. If your blood type B, that means the plasma be anti-A, B antigens, you can accept B and O. If your blood type AB, you can see how you have both antigens on the outside, have no antibodies, because you have both antigens, and you are the universal recipient. You can take any type of cell because you don't have any antibodies. If you're O, you don't have any antigens, so this is the universal donor. Like you can give this to anyone and it goes undetected. There's no antigens, but you contain antibodies in plasma, anti-A and anti-B. So you're only compatible with O, but you're a wonderful person because you can give your blood type to anyone. So you should really give blood if you're O, especially if you're O negative. This was the same picture as before, but I just want, now that we understand the antibodies in the serum that they give, let's say this person was like A, right? This is the blood. When they give them anti-A, that's antibodies A, or A antibodies, and then it agglutinates because they agglutinate with one another. So what are the percentages? If you're O positive, and A positive, they're the most, they're the most abundant of people. O negative, A negative, these are all. AB negative is like the least. Here's just another, the allele frequency. So your alleles are just the O, A, and B. So if you are American, there's a higher frequency, so there's more percentage of people that are O. French have more of A, but I mean, it's O again. The interesting one is Hindu. Those people are, that group of people have a higher percentage of B than anywhere else. So it's interesting to see in different places of the world where the frequency is higher than others. But really, universally, we all started as O. So RH blood groups, most Americans, 85% of us are positive, RH positive. So not only do you have an antigen that is AB or A and B, you also will have an RH. So if you're RH positive, that means you have the RH antigen. If you're RH negative, it means you do not have the RH antigen. So unlike ABO system antibodies, the RH antibodies are not formed at infancy. So you're born with your other antibodies. So I'm type O, for instance. So I was born with A antibodies and B antibodies. But I'm O negative. So I do not have Rh antibodies unless I'm exposed to the Rh. Then you create antibodies. So, and that's just like everything else. Most sicknesses that you get, you don't get antibodies until you're exposed first. And the same with Rh. The only antibodies you're born with are ABOs. Or I'm sorry, just ABs. So, hemolysis, this is the rupture of the red blood cells, does not occur when the first transfusion happens. So we're just talking about Rh now. So you have a transfusion, you're negative, and you're given positive. So you're given this positive Rh. It takes a time, so you get exposed, nothing's going to happen, you're just going to make antibodies. So if you get exposed again, now things are going to be problematic. So the second time and every time thereafter, the patient's antibodies attack and will rupture the donor's Rh positive. So this is what's going to occur. So then you again, remember, you'll just have that same thing that will occur with the kidney failure. So why would you get exposed more than once? If you're pregnant. So if you're negative and you get pregnant, and let's say your partner is positive, so therefore your baby is positive, that's when you would be exposed more than once. So that's what we're going to talk about. But first, let's go into this. So if you're O negative, that means you have absolutely no antigens. A negative, just A, no Rh. Negative means you're lacking of the Rh antigen. B negative, just B, no Rh, right? Negative means none. AB negative means you have A and B, but no Rh. Oops, sorry. Now you can kind of see, and I really like this diagram, that's why I put it underneath the other one, but O positive, you don't have A or B, right? You're O, but you do have the Rh, you're positive. A positive means you have both A and Rh. B positive, you have both B and Rh, and AB positive, AB and Rh, you have all the antigens. So you are the official recipient. So this is the universal donor, because there's absolutely no antigens whatsoever, and this is the universal recipient, AB positive, because you have every antigen. So your body would recognize every antigen. So this will explain the problem. So in Rh pregnant women who carry Rh positive babies, the first pregnancy usually results in a healthy baby. But now you've been exposed, so your body will produce these antibodies. And then this will lead to like probably miscarriage. You wouldn't even get past anything for the mother to be affected. You just time and time again would not get pregnant. You would miscarry very early on because the body would attack the baby. So in order to keep this from happening, they give the mother Rogam. So for instance, when I was pregnant with my son, they knew that I was what my blood type was. So they had it ready. So Rogam is an immune serum, must be given immediately after giving birth. So if the person is negative and their partner is positive, then when they give birth to their baby, they just have it ready and they just give it to the mother, even whatever the baby is. So that the next time that if the baby was positive and gave them antibodies, it wouldn't affect them. So really, it doesn't affect you till the baby is born. So here, this is a really good example. So mom is negative, dad is positive. So planets align and the baby is also positive. So while she's pregnant with the baby, it starts to enter the mother's bloodstream, but it really doesn't, the antibodies don't really form until after the baby is born. So once the baby is born, she would make these antibodies. So then the next baby was born, it would attack the baby when it was positive again. But instead they give them Rogam, and then they don't produce any antibodies. So then they'll be fine after that. Okay, and that's it. Perfect timing. So we will definitely practice blood typing, Punnett squares, and all this stuff this week. And we have a great lab as well to go along with it. That's great. I'll see you in a little bit.